As the body ages and gains excess fat, a key shared casualty is mitochondrial function, the ability of cells to efficiently convert fuel into usable energy. Mitochondria, often described as the powerhouses of the cell, are essential for maintaining metabolism, physical vitality, and even healthy cellular signaling. But in both aging and obesity, these tiny structures become less efficient, more damaged, and fewer in number. This decline is not a minor or isolated issue. It underlies many of the fatigue-related complaints common in older adults and those struggling with weight gain, and it contributes directly to fat accumulation, insulin resistance, and the gradual erosion of physical and mental resilience. In healthy young cells, mitochondria produce ADP, the body's main energy currency, through a process known as oxidative phosphorylation. This process is highly efficient when the mitochondria are functioning properly, enabling muscles to contract, organs to function, and the brain to stay sharp. However, as people age, mitochondrial DNA accumulates damage from oxidative stress, environmental toxins, and simply the wear and tear of time. Mitochondria begin to produce less energy and generate more free radicals in the process. This self-amplifying cycle of damage not only reduces energy availability, but also promotes chronic inflammation and accelerates cellular aging. Obesity adds another layer of dysfunction. Excess fat, particularly visceral and ectopic fat stored in muscles and organs, impairs mitochondrial health by increasing oxidative stress and disrupting the balance of energy intake versus expenditure. Fatty acids flood cells in large quantities, overwhelming mitochondrial capacity and leading to incomplete fat oxidation. This inefficiency promotes the storage of additional fat and the generation of harmful byproducts, such as reactive oxygen species, ROS, which further damage mitochondrial membranes and DNA. In obese individuals, mitochondria are often larger but fewer in number, less mobile, and less able to adapt to changing energy needs. This contributes to the characteristic metabolic inflexibility seen in obesity, where the body struggles to switch between burning glucose and fat effectively. The consequences of mitochondrial decline in both aging and obesity are wide-ranging. At the most basic level, it leads to reduced energy availability, which manifests as persistent fatigue, slower recovery after activity, and a greater sense of exertion during exercise. This can discourage physical activity, leading to further deconditioning and fat gain, a cycle that becomes increasingly difficult to break. On a deeper level, mitochondrial dysfunction contributes to the development of insulin resistance as poorly functioning mitochondria impair glucose uptake and storage. This, in turn, elevates blood sugar levels and pushes the body further into a fat-storing state. Cognitive effects also emerge. The brain is one of the most energy-demanding organs in the body, and reduced mitochondrial output can impair memory, concentration, and mood. In fact, mitochondrial decline is increasingly recognized as a contributor to neurodegenerative diseases, including Alzheimer's and Parkinson's, particularly when paired with the metabolic strain of obesity. Mitochondria also play a role in regulating cell death, apoptosis, which, when disrupted, can increase the risk of cancer or lead to the accumulation of dysfunctional cells that promote inflammation and tissue breakdown. Despite these grim effects, mitochondrial health is remarkably responsive to lifestyle changes. Physical activity is one of the most powerful stimulators of mitochondrial biogenesis, the process by which new mitochondria are created within cells. Endurance and resistance training both increase mitochondrial density and efficiency, improve metabolic flexibility, and help cells clear out damaged mitochondrial components through a process called mitophagy. Even short bouts of moderate exercise can significantly improve mitochondrial function in older adults and those with obesity. Nutrition also plays a critical role. Diets rich in antioxidants, from colorful fruits and vegetables, healthy fats, especially omega-3s, and polyphenol, found in foods like berries, olive oil, green tea, and dark chocolate, help protect mitochondria from oxidative damage. Certain micronutrients such as B vitamins, magnesium, and coenzyme Q10 are essential cofactors for mitochondrial energy production and may become deficient with age or poor dietary intake. Limiting highly processed foods, excess sugars, and trans fats helps reduce mitochondrial stress and improves cellular metabolism. Intermittent fasting or time-restricted eating, when done safely, has also been shown to stimulate mitochondrial repair pathways and increase energy efficiency. Sleep and circadian rhythm alignment are less obvious, but critical components of mitochondrial function. Poor sleep quality reduces the expression of genes involved in mitochondrial maintenance, while circadian misalignment, from irregular sleep patterns or late-night eating, disrupts cellular energy rhythms. Consistent sleep schedules and exposure to natural light during the day help regulate mitochondrial efficiency and hormone balance, particularly in older individuals. In sum, mitochondrial dysfunction is a key biological link between aging and obesity, a silent but potent force driving fatigue, weight gain, and metabolic decline. Yet the mitochondria are not passive victims of time or fat accumulation. They are dynamic and responsive to change. Through targeted movement, nutrient-dense eating, stress reduction, and circadian alignment, people can support mitochondrial regeneration and restore energy at the cellular level. This makes improving mitochondrial health not only a path to weight management, but a strategy for better aging, one that enhances vitality, protects the brain, and extends both lifespan and health span.